So the threshold for this paper is 32 out of 40, and let's just do it. An eyepiece reticule can be calibrated using a staged micrometer. What is the correct reason why an eyepiece reticule is calibrated? A is correct. When you do the practical, you literally just use it to measure stuff. Your eyepiece reticule can't be magnified. B is wrong. It doesn't magnify the specimen. And well, you use the measurements to make the comparison, not directly the eyepiece reticule. Typical eukaryotic cell. It's A, because eukaryotic cells, and you can see in the diagram there's no cell wall. There's only a cell membrane. There's only just one membrane going around. And B is wrong because it's a nucleus, not a vacuole. Don't see any chloroplasts. Nor do I see any lysosomes, so D is wrong. Oops. Which features are found in typical eukaryotes and in typical bacteria? Yeah, bacteria can respire. So it's definitely A, B, or C. Can't be D. Eukaryotes have mitochondria. So there's your circular DNA. Typical bacteria don't have 80S, only 70S. So A and B are wrong, so the answer is C. Which type of cell have the highest proportion of its volume taken out of cell structures bound by a single membrane? Well, single membrane will be the goblet cell, because goblets have a lot of lysosomes. Red blood cell doesn't have any cell structures, and if you know the structure of a companion cell, there are a lot of double membrane structures inside the companion cell. Same with the ciliated epithelial. The goblet cell mostly just has lysosomes because it's releasing mucus. That's why it's B. What causes the phosphate head of phospholipids to become polar? Well, the ability to form hydrogen bonds is because they are polar, not to become polar. Insoluble in water is just wrong. And D is wrong, covalent bond is wrong. But yeah, the reason they are polar is because they are able to ionize. That's the definition of being polar. So it's C. Which statement describes features of cellulose that adapt for its function in plants? Triple helix structure is wrong. 2 is correct. And you have your cellulose, it'll look like this with many hydrogen bonds between the parallel structures. Covalent bond is wrong because it's hydrogen as well. So automatically you know 2 and 3 can't be together, so they contradict. So A and C are wrong. And we know that 1 is wrong because of the triple helix. So it's D. Alpha glucose. So it can't be C. Has to have OH at the bottom. So it's B. Because when you see this, you know it's beta. So A and D are wrong. What cannot occur as a result of a condensation reaction? Breaking, because this is hydrolysis. By the time you condense, you produce H2O. So D is wrong. D is the direct result of condensation. And it's also between the two molecules joining together like this. If you're 1,4 or 1,6, so B and C are wrong. Which fact about the quaternary structure of proteins is correct? Doesn't necessarily need to be 4 polypeptides, could be more. Nope. This is just for hemoglobin with the iron. Depends on the primary structure. This is true for all structures actually, even the secondary and tertiary. So C is correct. Made of alpha and beta, not necessarily. Diagram shows different molecules in a solution. Which statement could explain what happens when some of the molecules are mixed together? So Q is the substrate, P is the enzyme. So A is wrong. It's not. It's the enzyme. This is the substrate, not the inhibitor. Molecule Q. Nope. Because this is talking about catalysts. None of these are catalysts. Oh, Q is not the catalyst. P is the biological catalyst. Molecules R and S bind to the active site of molecule P. Or D, molecules S and R are the products of the breakdown of molecule P. A little tricky, but C is more correct because you can see that these two parts are able to bind into the enzyme, so they can definitely stick to the active site. While Q, it doesn't necessarily need to come out as R and S, could maybe become like this. And R and S, if you look very closely, mix their shape together, it doesn't necessarily become Q shape, just looks kind of similar. Effect of substrate concentration on an enzyme catalyzed reaction was measured in 3 different conditions: without an inhibitor, just 1; competitive inhibitor, okay, let's just write out here, 2; and non-competitive is 3. Which row is correct? Oh, we already wrote it out, so let's just circle it. Which aspect of enzyme activity can be compared by the Michaelis-Menten constant, which is Km value? So basically it will be affinity, because Km relates to that. Won't be activation energy. So now, well, it won't be D as well. So now it's either B or C. So let's take a closer look. Km value will always be at 1/2 of the substrate concentration of Vmax, so it won't be C. But if you compare the affinity or the Km value between different enzymes and substrate, and if you can clearly see there's different values and different levels of affinity, so it's B. Number of substrate molecules can be blah, 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 blah. So how many times faster that converting substrate to product is catalase compared to phosphatase? So it's this and this. So let's just calculate this. So it's C. Red indicator solution was mixed with agar, and the resulting solid was cut into small cuboids. The blocks were placed in acid, which the indicator turns yellow. and all other variables were kept constant. The dimensions of the 3 blocks used are shown. So basically, let's break this question down. The ones with the highest SA to V ratio will have the least time to turn yellow. So A is wrong, just by seeing that. And D is wrong. So it's down to B and C, because they match that description. A way you can easily see without having to actually calculate it, if you assume that all blocks are the same side, same length, like this, they are all the same length. All of them are 3, 3, 3, 8, 8, 8, 11, 11, 11. When basically the side length increases, each side, so does the SA to volume ratio. So as you see, they are increasing, going from block 1 to block 3. So the SA to V ratio should increase. So the answer is B. Wait. Okay, let me just calculate this thing out. Okay, sorry about that. It's actually inverse. Your SA to V ratio will fall as the lengths increase. So the answer is C, because as the lengths increase from block 1 to block 3, you can see the SA to V ratio is falling. Sorry about that. Statements describe some events in the process of exocytosis. So, see, you're going to have something like this, SER, come a vesicle, and it'll move to the cell membrane, then it'll fuse, then it'll release whatever is inside. Okay, so 4 should be one of the first two, then 3 will be in the middle, and 2 will be the last one. So it's definitely C is wrong, A is wrong, because 2 is the last. Membrane of the Golgi body folds around the glycoprotein molecules. So Golgi body is going to take in the glycoprotein molecules first, then you'll form your vesicle, and then it'll move throughout the cytoplasm by the microtubules. Yeah, B looks correct. Okay. So basically, which had the least percentage change in mass? This is what the question is asking. So let's see. Because when your percentage change in mass is lower, that means your solution and your cylinder have similar water potentials. Let's say your cylinder has 100, your water potential outside is 1000. More will clearly go in, versus let's say I made it 10,000 now. Now there will be no net movement. You won't see a change in the mass. So by looking at this, you can understand that as the water potential difference is larger, you have more percentage change in mass, while if it's more similar, percentage change in mass will be lower. Contents of a daughter cell compared to the parent cell after one cell cycle. Okay. Number of chromosomes will always stay the same, so A and B are wrong. After a cell cycle, your telomere will shorten a bit, and volume of cytoplasm will reduce because I'm going from here to here. You're kind of losing your own cytoplasm as you're giving it to your child. So D is the answer. chromosomes are visible and lined up along the cell equator, so it looks like this. This is just metaphase. PMAT is PMAT. This is where they're condensing, where you lose your nuclear envelope. Metaphase is the lining up. Anaphase is where they're being pulled apart, and telophase is when they're gonna form. It goes from like this to something like this. They're gonna split apart. B is the answer. Which statements about the cell cycle are correct? Yeah. Yes. I got synthesis. And yeah, 3 is correct. So the answer is just A. So if you typically know that 1 and 2 are correct, you can narrow it down to A and B. Scientist stains the chromosome of plants with a fluorescent dye to observe the telomeres. Cell has 38 chromosomes. So 38. One chromosome will look like this. So there will be 152, take 38 times 4. But they mentioned it being in telophase. And in telophase, write out PMAT again. In anaphase, they already have been split apart. They look like this. And that carries on the telophase. So you need to take this and divide by 2. We will get 76. Answer is B. Doing a semi-conservative replication of DNA, double helix is unwound by an enzyme. The case. Which diagram shows how the enzymes are copied? Okay, so you know polymerase will act 5 to 3 prime. But let's see. Okay, lagging strand on here, definitely wrong. Because lagging strand will be the part with Okazaki fragments, where they are fragmented into different pieces. So A and C are wrong. Since you know your polymerase will act 5 prime to 3 prime, which means it will synthesize at the 5 prime to 3 prime direction. So this new one being made from 5 prime to 3 prime, this is the answer. Don't confuse it with polymerase reading this. It reads 3 to 5 prime, but it synthesizes 5 prime to 3 prime. That's what acting 5 prime to 3 prime means. So D is the answer. Transcription error results in the deletion of one nucleotide from the middle of a primary transcript. RNA forms from the primary transcript. Okay. A is wrong. It's not necessarily that whatever is replacing the deletion will still result in the same amino acid. It's possible but not guaranteed. Yeah, B is wrong. You'll definitely be affected. You're changing your primary structure. C is correct, cause as we stated before in one of the questions, your secondary, tertiary, and quaternary are all affected by a primary structure. Deletion changes your primary structure. Oh wait. Actually, after reading D, sequence of amino acids in protein will be different as all codons from deletion onwards are changed. C is wrong actually. Same logic with A. Similar logic with A, not the same. Because after the deletion, it's possible that the amino acid that will be produced is the same. Because multiple things can code for the same one. Like, for example, GAC and GAT could code for the same thing. So if T were after C and C were deleted, it would still produce the same amino acid. So sorry about that. It's possible for the same amino acid to form. So C is wrong. Sequence of amino acids will be different as all the codons from division onwards are changed. That is correct. So let's say, for example, JQGF is an example. Q is deleted, it will become JGF as a codon, so the sequence is now different. Nucleotide sequence on an mRNA molecule is used to produce a specific amino acid change. Yeah. Section of DNA is copied into an mRNA molecule by RNA polymerase. Okay, this statement is in fact correct, but only for transcription. It's mRNA, it's in transcription, and they're asking for translation. 2 is wrong. A polypeptide is produced because anticodons on tRNA molecules attached to mRNA molecules through peptide bonds. Peptide bond is incorrect because the peptide bond forms between the amino acids, not between the codons. So the answer is just 1. There were no introns present. Okay. The molecule of mRNA. So this is the mRNA. What is the sequence of the non-transcribed DNA for this section? Okay, let's write it out. So this is your mRNA. And okay, it will be C, T, A. So right now a lot of people would be tempted to just circle A as the answer, but that's not possible. Because this was the transcribed strand. And they are asking non-transcribed. Say a DNA looks like this, right? You transcribe this strand to get your mRNA. You're asking the leftover crap that you didn't transcribe. So it will be G, A, T. Won't be U because it's DNA. So it should be C. If you write everything out, you will get C. And this one, non-transcribed. Which properties of water are dependent on hydrogen bonding between water molecules? Cohesion, yes. Latent heat, yes. High specific, yes. So A is wrong because it doesn't have that. C is also wrong. D is also wrong. Answer is B. A lot of people get confused on solvent action, but even if you are unsure, just knowing the other 3 are correct, you can rule out A, C, and D as the wrong answers. But yeah, solvent action isn't really dependent on hydrogen bonding. Which substances in xylem tissue are impermeable to water and prevent collapse of the vessel? Now, preventing collapse is cellulose and lignin. There's a reason why cellulose has there's a reason for all the hydrogen bonds in the cellulose to make it strong. How impermeable to water is just lignin. Cellulose is permeable to water. Okay. These black things are not mitochondria, and A does not have that, so it's just wrong. And your sieve plates, yeah, okay. Donkey is wrong, C is correct. Carrier proteins in the cell surface membrane of companion cells are involved in the transfer of assimilates to phloem sieve tubes. The diagram represents the use two types of carrier protein in the process. So you're going to be pumping out your X. You're going to actively pump them out, and so X is your H plus. X is going to bind with Y, and they're going to be co-transported back. Y should be sucrose. Not glucose, sucrose. So B wrong. 28A. Okay, so which indicates atrial systole? Well, you know this dotted line, the one I'm outlining here, is your ventricle pressure. So anything going along there is wrong. So A is not possible, nor is D. And systole is when you're releasing that blood, right? Let's say I have this valve. Let's say this is a valve, right? These are blood cells. They need to pass through. Pretend it's a door. You need a lot of pressure and force to push open this door, to force it open before all of them can rush out. So in systole, you need high pressure. So in atrial systole, the pressure here in the atrium will be higher than the one in the ventricle. That's why your valve is able to be opened and send out all the blood into the ventricle. So for atrial systole, you gotta look for where the atrium has higher pressure than the ventricle. And at this point, B fits that description, while C definitely not. Ventricle pressure is way higher. An irregular heartbeat may be the result of ineffective electrical stimulation from the atria. Which area of the heart could be damaged, causing this irregular heartbeat? Can't be the septum or the node. Okay, so basically it's SAN, because the SAN is the one that is sending out the electrical stimulation. Let's say it used to send out 1 every 0.1 second to cause heartbeat, but now it's 0.1. Next one should be 0.1, but does it at 0.5, then goes back to 0.1, then 0.2, causing an irregular heartbeat. Well, septum doesn't really do much. Avian is just a place where it stops by, and Purkinje tissue only carries the stimulus and causes the beating of the ventricle. So it's just D, because D is, the SAN is basically the root of electrical stimulation. So when they mention ineffective electrical stimulation, it will be SAN. Which effect does increasing CO2 have on hemoglobin? So the more abundant something is, the more affinity there will be. So affinity for oxygen will fall, knowing that. C and D are wrong. And when affinity of oxygen is basically falling, it will release it more easily. So yeah, it will be B. Looking at this graph, the graph's kind of useless. Okay, but you can see that saturation of hemoglobin is with oxygen is like falling. which reactions will be taking place in blood in a capillary that is next to an alveolus. So you have your deoxygenated blood going to your lungs, which the capillary is here. So this is your deoxygenated blood. You're going to release your CO2 and obtain your O2. So 1 looks correct. 2 is wrong. You're releasing your CO2. You're not taking any more in. And since 2 is wrong, you can't have 3. So it's just B. Which structure of the gas exchange system always contains cartilage? Capillary is definitely wrong. Alveoli and bronchi are wrong. It's bronchus. If you look at it like this, your little alveoli here, the higher up you go, the more cartilage there is. So yes, trachea has your cartilage. And then your bronchus has your cartilage. So knowing this, alveoli and bronchial won't be the one with cartilage. It will be bronchus, because bronchus is higher up. But then typically bronchial and alveoli already don't have any cartilage. Exchange of carbon dioxide and oxygen occurs between air in the alveoli and blood in the capillaries of the lung. Which partial pressures of the gases allow gaseous exchange to occur? So basically you want, like a previous question, just very recently, CO2 is more in the blood and less in the lung. Okay, so let's take a look. So C is wrong, D is also wrong because there's less CO2 in the blood and more in the lung. And you also need to have more O2 in lung than O2 in blood. So it's B. Which labeled tissue prevents trachea from collapsing? So it's just cartilage. It's A, because you can see this little thingy here. It's a piece of cartilage. Which layers of cells does an oxygen molecule diffuse through when moving from alveolus into an alveolar capillary? So capillary will have your one cell thick endothelium. An alveolus will have your big, the alveolus looks like this, right? Big, flat, squamous epithelium. Also by common sense, you kind of can't have two of the same. So D and A are already wrong. Bacteria may be classified according to differences in cell wall structure. The antibiotic kills bacteria by inhibiting the synthesis of cell wall during growth. Which type of bacteria will be killed by penicillin more easily and why? It's gonna be the gram-positive one. Just cause, well, the thing is gonna attack the peptidoglycan layer, right? And in gram-positive, the first layer is legit just the peptidoglycan layer. While in the negative, it's the third layer. Penicillin needs to pass this and this if it wants to damage the peptidoglycan layer. So yeah, it just looks like A. Which facts relate to the virus TB or its pathogen? Nope. TB only has one type of antigen. TB... there's like multiple strains of Mycobacterium, so 2 is wrong. So you can cut out A, B, and C, so D is automatically the answer. HIV/AIDS makes the bacterial infection worse. It's common sense. Ingestion, yes. It's mad cow disease. If you eat a cow infected with it, you're gonna get it. Yes, Mycobacterium bovis, the thing is literally transmitted from cows. And yeah, 6 is possible in bacteria. Because the thing that causes TB is bacteria. Events listed occur during the primary immune response to a specific pathogen. Two is going to be one of the earlier ones, and 1 is going to be one of the later ones. 3 is going to be in the middle. 4 is going to be an earlier one, and 5 is going to be one of the later ones. Okay, so a phagocyte is going to consume the pathogen. A macrophage is going to kill it. So this is going to be the first one. Then after that, you have your expression. Then this will be third, fourth, and fifth. Okay. Yeah, lines up with B. The answer for this is A. It's like kind of just common sense. When you have an untreated HIV infection, it's going to be easier for you to contract more diseases. When your immune system is weak and has fewer memory cells, you'll be less able to combat the second coming of influenza. And when you have different antigens because of a mutation, you can basically assume it's like a completely new virus that requires you to go through the same process again. Your current memory cells aren't going to be able to help fight against the influenza, so that's why there will be a second strain. So thank you for watching till the end, and comment below what other past year papers you would like me to do. Thanks.